So I'm going to share my screen. Thank you so much, Natalie. I'm very excited to have the opportunity to talk to everyone today. Can you see my slide?
Yes.
Okay, wonderful. So yeah, so we're very excited in my lab about using different tools to investigate what's under the hood of cells, if you will, basically how cells are organized. And I'm talking about eukaryotic cells, which make up a large swath of life on Earth. And what I think is really fascinating about these eukaryotic cells is that they're all similarly subcompartmentalized into 9 different membrane-bound compartments. There's also all kinds of other cytoskeletal structures, and they're membraneless organelles. But one of the things that has really been important for our understanding of the way that the cell is functioning is being able to not only identify these organelles within cells, but to characterize their behavior relative to each other. And that has become extremely powerful over the last, I'd say, 15 years or so because of imaging technologies that have been implemented really since the really pioneering sort of recognition that you can introduce fluorescent proteins into cells using GFP and its daughters or cousins. Now, there's basically 2 different ways to look at organelles if you are imaging cells. One is a static modality, electron microscopy, and the other is light microscopy. And both of these approaches are really complement each other in many different ways in terms of really allowing us to understand how these organelles distribute, interact with each other, and function within cells. So today I want to share with you some new innovations in these 2 different
that's the horror.
In these 2 different domains. In the case of electron microscopy, I'm going to be talking about the whole volume focused ion beam scanning electron microscopy that's recently been developed at Janelia as well as elsewhere, where basically you can look at whole volumes, huge volumes at isotropic resolution, typically between 4 and 8 nanometer voxels to look through a whole specimen. And it's super exciting in terms of being able to put together all of these organelles as well as cells in these large volumes. In the light microscopy realm, there's also been many innovations from super resolution imaging to sort of live cell sort of multispectral imaging. I'm going to be talking about several of these technologies. The point that I want to make at the outset is that the information that you get is incredibly complementary from what you would get with electron microscopy, in the sense that you get global contrast or context for things happening within the cell. You have the ability with light microscopy with the fluorescent proteins to obtain protein specificity. And finally, you can look at organelle dynamics and interactions at incredible speeds and spatial resolutions. So let's start with the innovations in electron microscopy, this whole cell volumetric FibSim that I was talking about. Basically, in this approach, a specimen is basically high pressure frozen and put into a block, a tissue block, if you will, and then sequentially imaged by scanning EM and focused ion beam milling. The scanning EM allows you to get the XY, very fine XY resolution, and the fib-sim mills off different parts of small parts of the tissue to allow you to reconstruct at fine Z resolution the whole three-dimensional organization of the object that you're looking at. So it's isotropic, large volumes. But what's also exciting about this approach is that you can introduce a correlative step where before you acquire your fib-sim images, you can take a snapshot of fluorescent proteins that have been introduced into your specimen, and then you can superimpose that image over your electron your scanning EM image, and thereby get a sort of protein specificity to your image. So here's a pipeline that we've been using, and this was really developed by Harold Hess, and together with Eric Betzik. Eric really pushed the cryo implementation step, that basically cells are put on, we plate cells on sapphire cover slips. They are then high pressure frozen. And this is an important step because unlike the typical transmission EM approaches that people use to look at cells by EM, which use chemical fixatives, there's no chemical fixation. Basically, cells are very rapidly frozen in their near native state, and they are kept there throughout this whole process. So how do you image cells that are expressing a fluorescent protein that have been high pressure frozen? Well, you've got to make a microscope that can image at low temperatures, and that's what Harold and Eric did. Basically, they created a microscope where basically you can take your specimen right out of the high pressure frozen step, put it into this cryo, either structural elimination microscope, or we can also use PALM, single molecule imaging, acquire the image that we're interested in to get protein specificity. In this case, you're looking at the ER and a Cas7 cell. And then from that, the cells are moved into a state where we free substitute to get EM contrast. They're then resin embedded. We identify the cells that we're interested in for doing fib-sim. And then basically we move through a cycle of fib-sim trimming, milling, and imaging block face SEM, or acquiring the block face SEM images. So what do you see? What do you get when you do this? And here's an example of a HeLa cell where basically the entire cell has been sectioned through at 4 nanometer resolution. So we're getting isotropic, basically X, Y, and Z are all the same in terms of their resolution for about 4 nanometers. This particular HeLa cell, it took us 2 weeks to fib-sim through it. And that was possible because Sean Zhu at Janelia has developed basically he's tuned the fib-sim microscopes so that they work seamlessly over many, many weeks. And that's really important because the initial tries at this, basically the fib-sim milling, basically the machine would stop. And then when it restarted, you would be in a different position, which makes reconstruction extremely difficult. So it's really important to have these microscopes fine-tuned so that they can do serial milling over the course of days, weeks, if not months. So this basically took two weeks to acquire this data. And here's just some of the information that you can get from it. This is correlative cryo-PALM fib-sim imaging using fluorescent dyes. This is the first example of this cryo correlative fib-sim approach from Eric and Harold. Basically, this is a PALM image in the lower left in B, where you can see a mitochondrial probe, TOM20, superimposed with an M-emerald ER marker. And you can see beautiful identification of the ER and mitochondria in these images. So this is really very cool to be able to see all of these organelles identified through this type of labeling. It turns out that we've realized that you can also image various metabolites and other types of properties of these organelles using fluorescent dyes, which also are preserved in their fluorescence at cryo temperatures. So here's some work from Andy Moore in my lab where basically he's using basically we found they're cryostable. TMRM, which is a mitochondrial membrane potential dye, and Cyber Green, which is a mitochondrial DNA marker. And if we imaged, and this is a basically cryo structural illumination microscopy, we snap an image of the two different fluorescent probes. They dock right into mitochondria, which we can see through the FibSim reconstruction. So there's, I think, a lot of potential for this correlative approach to begin really characterizing properties of these organelles that we had not previously appreciated. So the correlative allows you to not only identify the organelles, but if you start introducing other probes, you can start characterizing, in this case, in the case of mitochondria, where the DNA is, where's differential membrane potential. We can also look at the plethora of organelles within cells from these FibSim data sets without any label. And so here, what you're looking at is a cubic volume, basically that's been sectioned through. You can see the centrosome in the center, and then all of the organelles listed to the left are color-coded and displayed in this cubic micron volume, so that you can look at all of these organelles, looking at their relationship relative to each other, etc. Now, this is pretty exciting. This was all done by hand by a single person, and it took them about 7 to 10 days. They're expert at doing this, but just to basically segment out one square cubic micron took 7 to 10 days for this person. And so we estimated that it would take about 50 years for that person to go through an entire HeLa cell, which is about 7,000 cubic microns in its dimensions. So that's definitely not something that is doable, and basically has led us, as well as many others in the field, to realize we need to, in some way, speed this up. And the way to speed it up is using computational approaches. So at Janelia, we've set up a team of researchers where basically we're acquiring the EM data using the FibSim tools that I mentioned. We're then teaming with computational people who are using machine learning algorithms to try to automatically segment the organelles that we know exist within these cells. So basically, here's the pipeline that we use when we try to automatically segment across an entire cell. The first step is to basically process, get all the data off the machine so that it can then be used by annotators to take select areas of the cell to segment by hand. Those annotations, which represent maybe less than 1% of all of the volume of the cell, are then used as ground truth by machine learning computational people to then see if they can basically teach the computer to identify the organelles that we've clearly been able to recognize, to do this automatically. And once that's done, there's an evaluation process where the computational people are speaking to the annotators to make sure that what the machine has done is correct. And that's a whole process of going back and forth. And then once that's satisfied, basically the next step is processing this data and analyzing this data for interesting features that pop up when we compare different organelles, especially between different cell types. And then ultimately, a key part of this whole process is data sharing, because the data that comes out of this is so humongous that we don't know what to do with it. We can analyze certain things, but basically there's lots of people all around the world that will find a lot of interesting information in this data that they can mine themselves through data sharing. So here's just an example of a different HeLa cell that has been segmented, and then basically that's been fib-simmed, and then the organelles are automatically segmented through machine learning. Now, what you can see from this data set is we can look at basically we've identified 32 different organelles by hand that the machine learning can reproduce in these cells. Here's data from 4 different cells that have been analyzed in this way. And I'm just showing a comparison of the different sizes of these different organelles, including the nucleus, the ER, endosomes, Golgi, the abundance of ribosomes, microtubules, etc. Now, here's another example of what pops out from this. Here is the entire Golgi apparatus within a HeLa cell. This was something that I was particularly interested in. In red are vesicles that are surrounding that Golgi. But what I think you can see in viewing this organelle is that it is a huge structure that's spanning many microns in the vicinity of the Golgi, of the nucleus. But all of its ribbon-like components are stacked, very tightly stacked, which we've known for many years. But I think what has not been appreciated is how all of these different stacked units, if you will, are interconnected with each other through tubule and other types of cisternal interactions. And so it's going to be very interesting moving forward to dig deeper into the organization of this structure using these types of approaches. Here's another example of information that comes out of this data. This is a single microtubule, and color-coded on its surface are different sites where different organelles are contacting that particular microtubule at that moment within the cell. And that was surprising to us because I never thought that so many organelles could attach to that small little 7-micron part of a microtubule. Here is the endoplasmic reticulum in a portion of the cell, and the COSEM team have basically outlined different parts of that microtubule. You can see the more smooth areas of that ER system are closely opposed to mitochondria, which are shown in gold. So basically, there is lots that can be done with these data sets, given prior understanding of these organelles. I mean, basically, if you know what the structure of an organelle is, you can easily identify it in these data sets. But there are lots of structures in cells that are hard to identify. And really, you're only going to identify it if you have the ability to correlate the structure with a particular protein of interest that can be used to define that structure. And so we've used this strategy to define an organelle, a structure, a compartment that we and many others in the field of membrane trafficking have been quite interested in for many years, and that's the ER to Golgi transport intermediates. What these structures do is deliver cargo that's newly synthesized from the ER into the secretory pathway by way of the Golgi. So we know from light microscopy studies many years ago that these ER exit sites are scattered all over the cytoplasm. Basically, they're scattered all along the surface of the ER, which is all over the cytoplasm. So they're emerging at specific sites on the surface of the ER, and then they basically bud off from that site and convey cargo into the Golgi, which is that central structure in this movie. So there's been huge interest in trying to understand what the ultrastructure of not only the ER exit sites are, but of the transport intermediates that are conveying material from that ER exit site to the Golgi. And so we decided that the FibSim reconstruction approach might be a really useful strategy for trying to get insight, better insight into this ultrastructure. So in order to do that, Aubrey and Qilun introduced a key part of the ER exit site, which is the COP2 coat. Basically, to identify these ER exit sites, we're going to introduce into cells a subunit of the COP2 coat, which drives budding out of the ER at ER exit sites, that's going to be our identifier for ER exit sites. So this is we're going to use this SEC23 subunit of the COP2 coat, and what you're looking at in this large left image here is the distribution of these COP2 ER exit sites scattered all over the cytoplasm. Now, what we're going to now do is zoom in on one of these ER exit sites. Basically, we're going to ultimately zoom in on all of them to look at their structure. But just as an example, what you can see is the overlay of the COP2 signal that we're getting from our cryocorrelative snapshot that's overlaid onto our FibSim images. So now we know where the membranes are that are going to comprise that ER exit site because it's got the protein that identifies these ER exit sites. So what do these things look like? And so here is here's where we've manually segmented that entire region that I showed you, or an example of it that I showed you in the prior slide. Now we know what the endoplasmic reticulum looks like in these cells because it's one continuous system. What we found, which was quite interesting, was that budding off from that endoplasmic reticulum was this elaborate tubular outgrowth. It's sort of like a grape-like cluster of membranes that the COP2 signal was honing onto. So this is a COP2 site, positive for COP2. You can see microtubules running on very parallel or very in nearby regions to this ER exit site. Basically, the entire structure has a diameter of about 360 nanometers, and the individual tubes are about 40 to 60 nanometers in this system. Now, we were able to show using photobleaching approaches, shifting over to light microscopy, that this is indeed a continuous structure. We can see it in our FibSim images. This red represents a neck. These things are continuous with each other, and we can see cargo moving in and out through photobleaching approaches. So we think this is one interconnected structure, the ER exit site, that's somehow differentiated from the rest of the ER sufficiently to sort cargo. Okay, so let's look at this cargo sorting. What's the effect on the structure of this ER exit site if we suddenly release a lot of cargo into it? And cargo, I mean secretory proteins that have been synthesized in the ER, and they're waiting to leave the ER to move to the Golgi and then ultimately to the plasma membrane. And so basically what we find, and this is FibSim data sets from either a control untransfected cell to the left of an ER exit site shown at the bottom, or of the same cell that we've transfected with TNF alpha, which is a cargo protein, and we've released into the secretory pathway using an assay called RUSH, developed by Frank Perez at the Curie. Basically, what you can see is 8 minutes after releasing this newly synthesized pool of proteins, TNF-alpha, into the secretory pathway, the whole structure grows. It bulges out. What's interesting is this: we don't get an increase in the overall number of these exit sites. Rather, we see that they basically just increase their size. So basically, the exit site diameter grows, and the tubule diameter also slightly grows. But otherwise, it remains still connected to the ER and has the other overall features of a ER exit site in a control cell. So that was really interesting. It suggests that this type of organization of these ER exit sites has the ability to transform in a conserved manner as a cargo, different types of cargo, and we show that in this paper, but also different cargo loads move through it. And that's a really sort of wonderful feature to sort of realize about the secretory pathway, that it can really adapt to different cargo loads, which is what it's going to have to do in different tissue contexts. Okay, so the next question, of course, is, well, what do the transport intermediates look like that move out of this system? If the ER exit site's still connected to the ER, what's happening? How do you form these transport intermediates that basically take things to the Golgi apparatus? And so for that, again, we did FibSim reconstruction to look at these transport intermediates. And so how do we identify these transport intermediates? We use this TNF-alpha rush cargo that we can release from the ER, and then we high pressure freeze 8 minutes after releasing that cargo, when we know there's going to be a lot of these molecules in ER to Golgi transport intermediates. So we high pressure freeze, no chemical fixation, and then we look at where is the TNF-alpha signal in these cells. And so we see some transport intermediates that are quite surprising in terms of the way they look. They're basically long purled membrane tubules. We're going to zoom in on this, one of these guys right here. So here's one of these purled membrane tubules that we can see the TNF-alpha SIM structural elimination light microscopy SIM image superimposed on top of. So we know it's got cargo. And moreover, this purled tube connects to an ER exit site. When we do our full 3D reconstruction, we see it connects to an ER exit site. This is the same image that I just showed in the movie. This transport intermediate connects to an ER exit site, and it's aligned along a microtubule. We can see all of this from this data set. Now, why have other people not seen this using sort of more traditional serial section transmission EM? The answer is they did not have the Z resolution. Basically, they had beautiful XY resolution, but the Z resolution was 40 to 80 nanometers. Our Z resolution is 4 nanometers here. So we get isotropic image. Plus, we're not just looking at one part of the cell, we're looking through the entire cell. So we can see these structures for the first time using this type of approach. Now, a question, of course, that sort of popped up is, well, is this just an artifact of overexpressing a particular cargo protein? Do other proteins do this as well? Under situations where, for instance, you're not transfecting anything? And the answer is yes. Now that we know what these structures look like, we know what an ER exit site looks like in these cells, we can come in and look at cells that have not been transfected with anything, and we can see the same structures by this FibSim reconstruction. So we think that these transport intermediates, these purling transport intermediates that are emerging from these ER exit sites are true transport intermediates that can carry you from ER into the Golgi region. They may not be the only ones. They are the predominant ones that we're seeing co-localized with the cargo that we've introduced. So here's our model for ER exit site ultrastructure organization that was only possible because of this whole volumetric reconstruction of the secretory pathway. Basically, we see this ER exit site that we're calling, which is this pearled membrane tubule structure that buds off from, well, that's sort of emerging from the endoplasmic reticulum. And you can see a stable neck sort of connecting these two domains. And then you see this long transport intermediate pulling off from this structure that's still connected to the ER, which we're calling ER exit sites. Now, I don't have time to go into where different machinery that we know from beautiful biochemical and genetic work drives the formation of this system. But basically in this paper in 2021, we were able to provide data that is consistent with basically the COP2 coat potentially being at this neck region, driving, stabilizing that neck, and COP1 working more peripherally to basically fine tune this whole transport intermediate, ultimately potentially retrieving things back, sort of basically streamlining this thing through some type of retrieval pathway. Now, for people in the field who are interested in these coat proteins, COP2 in particular is driven by a cycle of GTPase binding and dissociation. SAR1 is the GTPase, which when it's in its active GTP-bound form, will recruit the COP2 coat onto membranes. And so basically in yeast and other biochemical assays, the COP2 coat has been shown to be able to bud vesicles off of membranes. And that's really created the whole foundation of our thinking of vesicles budding from ER to Golgi and Golgi to plasmembrane, et cetera. What our data is suggesting is that this COP2 cycle of binding and dissociation could be coupled to a membrane sorting process that, if it's robust enough, basically leads to the creation of this elaborate membrane outgrowth that the continual binding and dissociation cycle of COP2 would stabilize. And then ultimately, this thing here starts differentiating through recruitment of different phosphoinositides, cholesterol, you name it, and it starts engaging in a whole different sort of pathway of behavior relative to the rest of the ER. So that's what we're thinking. But basically, what this data is showing is that using whole cell cryocorrelative sim fib-sim provides a new approach for understanding different pathways within cells that, although we're really ready for this at this point because we know a lot about the biochemistry that's driving these membrane differentiation events in these different pathways. What we don't have a grasp yet on is exactly how those biochemical pathways are linking to the actual ultrastructure of these membranes in whole cells. So I now want to shift for the last 10 minutes of my talk to interrogating organelle structure dynamics by light microscopy. As I mentioned at the beginning of my talk, this approach provides some stuff that you can't get from EM, like dynamics. Basically, these organelles are moving all over the place, and that movement is important for positioning the organelle to a place where it can contact and communicate with other organelles or be exposed to particular cytoplasmic environments. So we need to think also about dynamism. And so I want to share with you some work that we've been doing in this front. one of the things that we did many years ago was to really demonstrate that you can definitely look at many organelles simultaneously by introducing different fluorophores to different organelles. In this case, we have 6 different fluorophores targeted to each of these different organelles. Normally, the fluorophores, if you tried to image them, would overlap in their spectrum if you're just using conventional imaging, but we can use spectral unmixing to distinguish each of these structures. And if we image with a lattice light sheet, we can also get somewhat isotropic visualization of these organelles to allow us to be able to segment them and look at their diffraction limited association with each other. Basically looking at how frequently different organelles are coming closely opposed to each other. And from that analysis, we were able to really hone in on 2 organelles, ER and mitochondria, as really a hotspot for inter-organelle communication. And this is illustrated here, where we've just taken that lattice light sheet data, and now we're going to just segment out all of the mitochondria to the left, and then we're going to look at what portion of that mitochondria we can see ER in close association with at any particular time. And that is what you're looking at in the right image. So basically all of the green yellow signal that you're seeing on top of that mitochondria represents ER that is very closely associating with the mitochondria over this time period. So what is the ER and mitochondria doing at those closely opposed regions? There's a whole field that is interested in this and has shown with beautiful assays that there's a lot happening at these contact sites. They are not fusing. They are staying right next to each other because tethering proteins stabilize them. And as a consequence of particular proteins getting concentrated there, you can get lipids transferred between ER and mitochondria. You can get calcium flux, you get ROS release, and these mitochondrial ER contact sites have been shown to be involved in division of mitochondria. So can we look at these contact sites and analyze them in more detail? Now, I just showed you FibSim can definitely find these contact sites. And this is just an example of a contact site in FibSim. Basically, we can see the whole 3-dimensional structure. The red is that contact site where they're within just a few nanometers of each other, these 2 organelles. And what I think you can see is that where the ER and mitochondria are contacting, it's sort of a flat interface. Well, this is a static image, and we don't have any protein specificity associated with. So we don't know what proteins are localized here, and we don't know what those proteins are doing and how their tethering proteins are actually stabilizing this. So basically, to address this, 2 people in my lab, Chris Obera and Johnny Nixon-Abel, who's now has been at Cambridge for several years, they developed an assay to see whether they could characterize the proteins that are associated with these ER mitochondrial contact sites. So this is single molecule tracking. And I think the innovation that Chris and Johnny brought to the table here was basically being able to superimpose the trajectories of individual proteins within the ER, which you're seeing in green, on top of the organelle itself, the ER itself. And that's really important if you're going to try to understand really what's going on with these molecules. So in order to sort of focus in on the behavior of particular types of ER proteins, Johnny and Chris looked at this molecule called VAP-B, which is a tethering protein that's been identified to tether the ER to different organelles. And in the case of mitochondria, it tethers it through interacting with this protein P51. So basically, they form a tether that holds these 2 organelles close very close to each other so that they can engage in interorganelle communication. So here is a movie showing a halo tag VAP-B, where we're also looking at the endoplasmic reticulum in white and the mitochondria in red. And what you could very quickly see from these live cell imaging data was that the VAP-B likes spending time around the mitochondria. So the VAP-B is only in the ER, but in areas where the ER is close to mitochondria, its behavior is changing. It's still moving, and it can move in and out of the regions of close association between ER and mitochondria, but it likes hanging out in that region. And this is illustrated here, where we're just summing up in different colors the different trajectories of VAP-B that we're acquiring. And you can see these little hot spots of where VAP-B is sort of changing its trajectory very, very quickly over time. So this top image that you're seeing here in white. This is in blue here and red, this is a single VAP-B trajectory along the surface of the ER. And what you can see is the molecule moves very, very fast when it's in an isolated tubule or, three-way junction, whatever, in the ER. But as soon as it gets to a region that's close to mitochondria, which we believe is a contact site, it undergoes very, very concentrated movement, retaining it in that structure for a time before it can move out. And so here's just an example where we're showing this is an outline of the mitochondria. Here is the endoplasmic reticulum. And what is highlighted by this arrow is one of these hotspots, which is basically what we've done is created a color coding of the VAP-B probability densities of all of the molecules that we're imaging. So there's a hotspot there, and it correlates with the behavior of these molecules. So here is a single VAP-B set of trajectory over several seconds. And you can see it's either free when it's coursing through the ER, or at sites where the ER is in close apposition. We think this is a contact site. The VAP-B is spending a lot of time. Okay, so that's pretty cool. It allows us to basically get single molecule super resolution of these contact sites. So here's mitochondria in red, in cyan is ER, and this probability density plot just shows you all along the surface of the mitochondria where the contact sites are. And this is specific for VAP-B. It's N-terminus. This is actually the region where VAP-B can interact with the mitochondrial partner. If we get rid of that region in this delta N VAP-B, you can now see there's no enhanced probability density of VAP-B. It's just diffusing all over the ER, and it doesn't correlate with anything associated with mitochondria. What's more, basically the size and shape of these contact sites that we're identifying using single particle tracking are very similar to what we see using FibSim. So in this left image here, this is a FibSim reconstruction of ER, and in red are ER mitochondrial contacts. I'm not showing the mitochondria in this image for the purpose of just looking at these contacts, but we know from the FibSim image, these are the contact sites. So we say, okay, well, let's take this region here and project it in 2D and look at what we would see. And what you're seeing at the bottom is just a projected 2D image of this FibSim reconstruction. This is where the contact sites are, and this is exactly how they would appear in 2D. And what's interesting is that's exactly what they look like in our single particle tracking, which is 2D. Basically, we're imaging, by single particle tracking. So we only are imaging in this system in a 2D dimension. So we have very similar overall shapes, which is very reassuring, and also is telling us that we have nanometric information on these contact sites. So what can we learn from this? We can start doing some interesting things like who's controlling the size of these contact sites. If we overexpress VAP-B, no change in their shape. But if we overexpress the mitochondrial partner, we get a huge change in these contact sites. And this is just showing the FIPS, our single particle probability density analysis. So basically, it's the mitochondrial partner that's driving the size of these contact sites. Well, we have single molecule information in these contact sites. We know what their shape is. Can we get information on the diffusional landscape within the contact sites of a particular molecule like VAP-B? And this is an attempt to do that. Basically, we're plotting all of the molecules that have moved in and out of this contact site that we've identified by this localization probability. And we can determine the point diffusion coefficient of the molecule at different positions across that whole contact site. And this is what you're looking at in this right-hand panel. This is the localization density of the VAP-B in the contact site. And here is changes in its diffusion coefficient as individual molecules move to different positions in that contact site. The point that I want to make is that there's a single low diffusion well. When the molecules get to the center of this contact site, they are diffusing the slowest. We can also see what happens when we create conditions that are known to change contact sites, like starvation. So when you nutrient starve cells, these contact sites change because basically you need more communication between ER and mitochondria under nutrient starvation conditions. And what we can see clearly from this analysis is that the contact sites not only get bigger, but they also change. The diffusional landscape of VAP-B changes across that contact site under these conditions, where molecules are still moving in and out, but when they get to the center of this contact site, they're undergoing much slower diffusion, which we think might relate to the tethering efficiency. Basically, the binding dissociation of these molecules seems to be changing as cells are nutrient starved. Well, one of the things that we were interested in looking at with these, with VAP-B in particular, is its pathogenic mutation, P56S, which is basically one of the mutations that can give rise to a neurological disorder, Lou Gehrig's disease, ALS. What happens to ER exit sites in cells where we express this mutant form of VAP-B? And to the right shows the diffusion map of a typical ER mitochondrial contact site under these conditions. And it's pretty striking in terms of how it's changed relative to the wild type situation. Basically, now we see multiple very low diffusion wells. And we now see for the first time that VAP-B molecules will go into this system and won't come out. Basically, they go into a diffusion well that's so slow that the molecules never come out over the course of our time-lapse imaging. So we think this is very significant in terms of the pathogenic features of this mutation. Basically, this data is suggesting that these contact sites are now becoming much more aberrant in terms of their tethering characteristics. And that might in turn impede their ability to be able to remodel under different conditions, like I just showed you with nutrient starvation. And that itself might lead to ultimate sort of metabolic dysfunction in these cells. So with that, I want to just recap the contact site data. Single molecule tracking of VAP-B is really not only allowing us to look at these ER mitochondrial contact sites for the first time at sort of super resolution, but we can see that the VAP-B molecules are moving out of these contact sites. They're not just going in and, like Velcro binding to a binding partner. The binding must be very dynamic. based on what we're seeing. And we can also see that these ER mitochondrial contact sites change their morphology and dynamics under different conditions like starvation, overexpression of the mitochondrial partner, PTPI51, and expression of the pathogenic BAP-B mutant. So with that, I want to just end and thank you for listening. I just want to emphasize how we're really at an incredibly exciting time in the imaging world, where we have at our fingertips many different technologies that allow us to interrogate at so many different levels intracellular organization, from structural information of a whole cell, as I mentioned with the FibSim reconstruction, to various live cell imaging technologies that improve both spatial and temporal dimensions. So with that, I want to thank all of the people who've made this work possible in the FibSim world. I really want to thank Harold Hess and his team, who've really made these microscopes made it possible to be able to do these huge volumetric reconstructions. I also want to thank Eric Betzig, who's really with Harold enabled the cryo implementation in that FibSim system that was so important, at least for our work, for looking at these ER to Golgi trafficking intermediates and the ER exit sites themselves. Single particle tracking, I really want to call out Chris O'Bara and Johnny Nixon Abel, who've really driven that project, looking at the ER molecules, but they've been helped by some really important players in analysis, including Jean-Baptiste Masson from Pasteur and Chris Calderon from Ursa Analytics. We're also very thankful with our tight communication with collaboration with Craig Blackstone, who's an ER expert. And then finally, I really want to thank the COSIM team, project team at Janelia, headed by Aubrey Beigel, who has really made the FibSim technology available to biologists like myself to really interrogate. And so with that, I'm going to stop, and I'd be very happy to take questions.